In this video, we'll talk about cell structures. We'll talk about the structures of bacteria, archaea, and eukaryotes. We'll talk about the innovations that evolved in eukaryotic cells. And as we discuss the complex features of eukaryotic cells, we will discuss what to look for in the wonderful Inner Life of the Cell video that shows how the various parts of the eukaryotic cell interact in a living cell. So let's start off with prokaryotic cells. Recall that prokaryotic cells do not have a nucleus, and these are bacteria, sometimes called eubacteria and archaea. Archaea are sometimes also called archibacteria. So the main feature of a prokaryotic cell is what it doesn't have. It doesn't have a nucleus. It has DNA, and the DNA is a circular chromosome. So the DNA, as noted here, is usually a single circular chromosome molecule. In addition, and it occupies a region of the cell called a nucleoid. There are other, or there can be other DNA molecules. They're also circular, but they're much smaller, and they're called plasmids, and we will discuss plasmids when we talk about genetics later on in this course. There aren't really much else in the features of a prokaryotic cell. Both bacteria and archaea have very simple cell architectures. Of course, there is a plasma membrane, and then outside of the plasma membrane, archaea and various branches of bacteria can differ in their cell walls and their outer capsules, but we're not going to go into those complexities. So in just looking at structures, it's very difficult to distinguish bacteria from archaea. They have similar cell size. They have, as we saw, very similar simple cell structures. Both of them have circular genomes. Both of them contain plasmids, and both types of cells divide by simple fission. What are some differences? Why do we classify them as being different domains? Well, as we saw before, their membrane phospholipids are very different. Archaea have these isoprenoid lipid chains, hooked up to glycerol through ether linkages, and bacteria have fatty acid lipid chains hooked up to glycerol through ester linkages. And this is also true of eukaryotes. When we look at their DNA, the sequences of the genes, what we see is that the bacterial and archaeal DNA are as different from each other as each is from eukaryotes. In fact, a closer examination shows that the ribosomes of archaea and their genes that are involved in DNA replication and transcription, those actually look more similar to the eukaryotic genes that are involved in DNA replication and transcription than they do the bacterial genes. So let's go on to cell structures, or let's go on to contrast prokaryotes with eukaryotes. One of the obvious differences is difference in cell size. Prokaryotes in general are smaller than eukaryotic cells. There is some overlap where the smallest eukaryotic cells are bigger than the largest prokaryotic cells, but in general, eukaryotic cells are much larger than prokaryotic cells, and eukaryotic cells can get very large indeed. at 1 millimeter, you can definitely see them by the naked eye. And of course, if you consider eggs, those are indeed just enormous eukaryotic cells. Eukaryotic cells have lots of complex internal structures. The defining feature of a eukaryotic cell is the nucleus, shown here in green. The nucleus is bordered by a double membrane that we call the nuclear envelope. And proceeding outward from the nucleus, of course, the nucleus has the cell's chromosomes, and I think this is a good time to note that in eukaryotes, the genome consists of multiple linear chromosomes. So all eukaryotic genomes known so far are divided into multiple linear chromosomes, which reside in the nucleus. So proceeding outward from the nucleus, what we see is that the nuclear envelope is actually contiguous with the endoplasmic reticulum, shown here. And what's shown here is the endoplasmic reticulum with ribosomes, which make them appear rough in an electron micrograph. And so we call that the rough endoplasmic reticulum. And then proceeding outward from there, the rough endoplasmic reticulum is contiguous with the endoplasmic reticulum that lacks ribosomes, therefore appears smooth in an electron micrograph, and we call that the smooth endoplasmic reticulum, or ER. If we look at other membranous structures within the cell, what we see is the prominent Golgi apparatus, or the Golgi, sometimes called the Golgi stacks. The Golgi stacks are part of what we call the endomembrane system, and I'll write that here. And the endomembrane system consists of the nuclear envelope plus the endoplasmic reticulum, both the rough and smooth, the Golgi vesicles, and the lysosomes. And finally, the endomembrane system is related or connected to the plasma membrane through secretory vesicles. As we will see, or as you will see in the Inner Life of the Cell video, there is continuous flow of membranes through the endomembrane system, where membranes and membrane proteins are generated in the rough ER. Smooth ER generates much of the lipids, and membranes form little vesicles that travel from the ER, fuse with the Golgi, and from the Golgi they form vesicles that either become lysosomes or become secretory vesicles that travel to the plasma membrane and fuse with the plasma membrane, expelling their contents to the outside of the cell. So that's one key area of emphasis that I would like you to remember and note, is the endomembrane system and what is in the endomembrane system. There are other membrane-enclosed organelles that are not part of the endomembrane system. The most important are the mitochondria. There are about, maybe 1,000 mitochondria or so per cell. Mitochondria, as I said, are independent organelles. They're not part of the endomembrane system. They have their own double membrane, and they have their own DNA. Other features of note here are an extensive cytoskeleton system, shown here as these sort of yellow ribbons, and centrioles, which are part of the cytoskeleton system, particularly the microtubules. If we go to the plant cell, all you need to remember about plant cells is that they are eukaryotic cells that have all of the exact same organelles as animal cells or any other eukaryotic cell, with the addition of chloroplasts. Now, we believe that most plant cells lack real lysosomes, and they also lack centrioles, but that's really the only difference, or the only major difference. So let's talk about how these three domains, the bacteria, the archaea, and the eukaryotes, how we classify them in relation to each other. So if we look just on the basis of structure, eukaryotes having nuclei and prokaryotes lacking nuclei, that is one way to divide, and that is the classic way to divide all of life on Earth, those with nuclei versus those without nuclei, and that makes a lot of sense. It's a very helpful division of life into eukaryotes versus prokaryotes. Now, if we start looking at genetic sequences, the picture is very different. A man named Carl Woese started sequencing ribosomal RNA genes from various species of bacteria, archaea, and eukaryotes. And what he discovered, to his surprise, is that based on these ribosomal RNA sequences, the ribosomal RNA sequences of the archaea were very different from the ribosomal RNA sequences of bacteria, and as different from bacteria as eukaryotes from bacteria. And in fact, the ribosomal RNA sequences of archaea actually were closer or more similar to the ribosomal RNA sequences of eukaryotes than they were to bacteria. So he realized that the only way the data fit in terms of a classification scheme was to have three separate domains, that bacteria and archaea comprise two major divisions of life, and eukarya constitute a third division of life, and he called these domains, and that the archaea domain and the eukarya domain were actually more closely related to each other than they were to bacteria. Now remember that this is simply based on ribosomal RNA sequences. That's only one gene out of the entire genome. It's a minuscule fraction of the genome. Now, as genome sequencing projects began to sequence entire genomes from bacteria and archaea and many genes in eukaryotes, evolutionary biologists were able to do deep comparisons of many groups of genes, and that allowed them to look further back in evolutionary time to virtually the beginning of life itself. And in fact, they were trying to figure out what the last universal common ancestor, the common ancestor of all three domains, the root of this tree of life, looked like. And what they realized was that they couldn't get a consistent tree. Some genes in eukaryotes look like they came from archaea. Some genes in eukaryotes look like they came from bacteria. There were some genes in archaea that looked like they could have come from bacteria, and so forth. And the only way to explain all of these inconsistencies was that early on in the history of life, the predecessors, the ancestors of bacteria, archaea, and eukarya, were actually happily swapping genes back and forth between each other. This is a process called lateral gene transfer. This is distinct from vertical gene transmission. Vertical gene transmission is when parents transmit their genes to their progeny. The normal, ordinary transmission of genetic information from one generation to the next. In this case, though, here we're talking about swapping bits of genomes from across species boundaries, not just between closely related species, but between species that are highly unrelated and may in fact be in different domains. So if you look at the base of the tree of life, what you see is a complex intertwining that reflects much exchange of genetic information. Today, with many fully sequenced genomes from all three domains, what we get is a picture that eukaryotic genomes evolved from a fusion, essentially, of archaebacterial and eubacterial genomes. Approximately half of eukaryotic genes, sort of the core eukaryotic genes that are found in all eukaryotes, have their closest relatives among the archaebacteria. The other half of the core eukaryotic gene set have closest relatives in the eubacteria, strongly implying that eukaryotic cells evolved through some kind of partnership or fusion event between eubacteria and archaebacteria. So here are the major hypotheses for how eukaryotes originated. So the prevailing hypothesis is for a fusion event or some kind of endosymbiotic partnership between an archaeon and a bacterial, ancestral bacterial cell. This could be either a bacterial host cell that acquired an archaeon as perhaps an endosymbiote, and the archaeon became the nucleus, and a reason to think that is because all the information processing genes appear to have come from archaea. So these are the genes that are involved in nuclear processes of DNA transcription, DNA replication. So the stuff that happens in the nucleus appear to be mostly of archaeal origins. And so it makes sense to think that maybe an archaeal cell became the nucleus. Whereas the genes that are involved in processes that occur out in the cytoplasm, such as energy metabolism, appear to have counterparts or close relatives among the bacteria. Another way to look at it is that we had an archaeal host that acquired mitochondria. So mitochondria, as we will see, has clear bacterial origins. So mitochondria came from bacteria, and that the half of the eukaryotic genome that appears to be bacterial in origin actually came from mitochondria, that the mitochondria shed their genes and gave them up to the archaeal host cell. The major alternative hypothesis to this fusion between archaeal and eubacterial genomes is that eukaryotes descended from a truly ancient proto-eukaryotic lineage of their own. But there are good reasons to think that eukaryotes evolved later than archaeal and bacterial lineages, and therefore most people favor some variant of this first hypothesis. Here's an illustration then of the role of endosymbiosis in the evolution and origin of eukaryotic cells. Here what's shown is some primitive prokaryotic host cell. It's not identified as either archaeal or bacterial, but it has prokaryotic-like qualities, meaning it doesn't have a nucleus. It probably had a circular DNA, single circular DNA genome, and had a very simple cell structure. What this shows is it acquiring mitochondria through a process called endocytosis. And in the process of endocytosis, the host cell membrane encloses the particle. So if the particle happens to be a bacterium, then what we have is the bacterium with its own plasma membrane surrounded by the host cell's membrane. And other invaginations, membrane invaginations, the plasma membrane making pockets into the cytoplasm, that's thought to have given, or that's thought to be the origin of the endomembrane system and of the membranes that enclose the nucleus. So membrane invaginations became the endomembrane system as well as the nuclear envelope. The bacteria then shed most of their genetic material. Most of their genetic material actually went into the nucleus and became part of the nuclear chromosomes. And what remained became today's mitochondria. Indeed, every eukaryotic cell contains mitochondria, and even parasites that have lost or that don't have mitochondria appear to have had mitochondria at one time before they took up their parasitic existence. So here we have our sort of ancestral eukaryote. Which then diversified into various protists, fungi, animals, or some eukaryotes acquired a second endosymbiont. These were photosynthetic bacteria, and just like the bacterial endosymbionts turned into mitochondria, these photosynthetic bacterial endosymbionts turned into chloroplasts, and these became photosynthetic eukaryotic algae and plants. So there is very solid evidence for the bacterial origins of mitochondria. So mitochondria, as I said, have their own circular DNA genome. And the sequence, the DNA sequence of the mitochondrial genome resembles most closely alpha proteobacteria. So we can even identify the group of bacteria that were the most likely ancestor of mitochondria. Similarly, for chloroplasts, chloroplasts have their own DNA as well, which forms a circular genome. And the chloroplast DNA sequence most closely resembles the DNA from cyanobacteria. Now, eukaryotes have these internal membranes, they have cytoskeletons, and they have the nucleus, which are not present in prokaryotes. How do these complex structures evolve? As people have looked into the diversity of bacteria, scientists have found that some groups of bacteria actually have some of these eukaryotic features. There are these two groups of bacteria that are related to each other that do have internal membrane compartments. Not only that, but these bacteria also have genes for endocytosis, which is quite rare among bacteria and archaea. And finally, they have genes to make some sterols. These are primitive sterols, not as evolved or as complex as eukaryotic sterols, but sterols nevertheless. This is evidence that at least these eukaryotic features didn't evolve in eukaryotes de novo, but possibly had, or possibly originally evolved in a group of bacteria. I'm not going to spend any time going into the complexity of the eukaryotic cell. I instead prefer to have you watch the Inner Life of the Cell video animation. It's a wonderful animation